Bismillahirrahmanirrahim. My name is Muhammad Nizam bin Lani from University Malaysia, Terengganu. I will present one of paper for 4th Asia Pacific Regional Conference on Food Security 2023 entitled Effect of Chili, Turmeric and Black Pepper on Microbiological Profile, Sensory Acceptability and Shelf Stability of Fermented Oreochromis niloticus at ambient temperature. This presentation will consist of introduction, materials, methods, result and discussion, and conclusion and suggestion. As we know, fish is most common source of animal protein that are prepared in many ways, including boiling, smoking, marinating, dried, and fermented. Fermentation is preferable in tropical countries due to the availability of carbohydrate sources, favorable condition for fermentation process, and it can be sold at a cheaper price. Fermented fish from freshwater fishes, including tilapia, is preferable than marine fish because the former has better flavor and texture. Salt is important in fish fermentation to prevent the activity of spoilage bacteria and allow fish enzyme and beneficial bacteria to break down the flesh. So normally at the industry, they use about 25% salt. However, the high level of salt use can lead to the hypertension and effect on consumer preference due to increasing health concern on the amount of salt taken. So therefore, in this study, we reduced the amount of salt into 20%. In addition, selected spices such as turmeric and ginger added in fermented fish have antimicrobial activity and potentially used as partial substitution of salt. For the problem statement, limited study on the effects of spices on the shelf life and the sensory acceptance of fermented fish products related to Percasam. There is also limited study in developing new low-salt fermented fish incorporated with the spices. For the justification of the study, local communities can be taught on this new innovated and healthy version of Percasam. Small and medium industry could utilize the local spices as flavoring agents in preparing fermented fish and allow a variety of tastes that could overcome the unpleasant smell of fermented fish. From this study, Department of Fisheries can acknowledge the new scientific data and information on the application of fish for the downstream products. The objective of this study to determine the microbial quality and lactic acid bacteria count of fermented tilapia incorporated with the selected spices. Secondly is to determine the shelf life of this product. And thirdly is to determine the sensory acceptance of the fermented fish. We go for the materials and methods. For the preparation of fermented fish, there are 3 spices were selected. So in this study, we preparing fermented fish without spices as control, and we also have a chili, turmeric, and black pepper, and at different concentration. And this will be studied for the shelf life study and microbiological quality, and also for the sensory acceptance. This is the tables describing the design of the study, where the commercial use about 25% for muddy, but in this study we are reducing the salt, and at the same time we have 3 different spices at 3 different concentration, 3%, 6%, and 9%. This is the table showing how the randomization block designs were conducted. This is the methods for the process of fermented tilapia. Basically, we are trying to find tilapia which had the weight around 300 to 350 gram from the local market in Terengganu. And after that, we need to gut it, clean and toast away and place directly in the ice water. And tilapia were rubbed with 20% of salt. And tilapia was left in the sterile container for overnight. And tilapia was mixed with 20% of crushed roasted rice, 5% of tamarind, and 5% of brown sugar. After that, the selected spices were minced traditionally by using sterile knife and cutting board. And the spices were mixed with tilapia at 3 different concentration. And tilapia was placed in sterile sealed and labeled plastic container, and it was fermented at ambient temperature for 50 days. After, for this study, the method used for the microbiological examination involved food homogeneous preparation, where tilapia fish were cut, heat, middle till region using a sterile knife, and 25 grams of fish steak cuts was homogenized in 225 ml of buffered pet water. And after that, the homogenate was added with the dilution blend, sterile buffered pet water. And then we perform the serial dilution. And because we are using a different microbiological quality parameter, there are various selective media were used. Plate count agar, potato dextrose agar, violet with bar agar, ethylene, eosin methylene blue agar, backpacker agar, demand rogosa and sharp agar added with 0.8% calcium carbonate. And the plates were incubated at the appropriate temperature, and the colonies performed after incubation were counted. This table shows the media that were used and the incubation period time and what other analyses were conducted for this study. Okay, after we did the experiment, some of the colonies we further studied for the gram staining. And then we do also catalyst tests because we want to ensure that the fermented fish contain lactic acid bacteria. As we know that lactic acid bacteria are positive, gram positive rod or cocci with negative catalyst test. And the last part of the experiment were for sensory evaluation, where we did for the acceptance test. Fish samples of 8 days of fermentation were mixed with their fermented roasted rice, and the sample were separately fried with 50 ml of oil per fermented fish, and fried fish sample were distributed to 30 untrained panelists to evaluate either they like or not the products. And these are the examples of the score sheet used in this study, where you're using a 7-point hedonic scale, where 7 shows the extremely like and 1 shows the extremely dislike. This is the experimental design used for in the study for determine the microbiology and lactic acid bacteria. This is the experimental design for the objective number 2. And then we go for the result and discussion. For the objective number 1, okay, this is the results showing the Staphylococcus aureus count in fermented tilapia. As aureus count in different percentage of added spices in fermented tilapia, we can see that there is the different letter A until D indicate the significantly different P at the less than 0.05. Histogram shows that mean S aureus count and error bar indicate the standard deviation. Okay, this is the results for enterobacterial count in fermented tilapia. We can see that the results are varies depending on the concentration of the spices. And from here, we can see there is some increasing and some decreasing of the enterobacterial count. And the figure 8 shows the results for the E. coli count in fermented tilapia. And it's also showing inconsistent of the reduction and of the effects of the spices. Okay, this is the results for the identification of lactic acid bacteria from the fermented fish. We can see that the lactic acid bacteria with the 3% black pepper in red color, 6% black pepper in the yellow color, 9% of the black pepper in green color. It shows that the LAB going up mostly for the first 5 days of fermentation, and after that the LAB decreasing. And it shows a similar trend for other spices, for turmeric and chilies. The confirmation of LAB were also used using the biochemical test, and we presumptively confirm that all the colonies from the plates in the MRS showing the gram positive with the cocci, and they are mostly catalyst negative, indicating that they are presumptive lactic acid bacteria. For the objective 2, to determine the shelf life of fermented tilapia incorporated with certain spices during ambient storage, from here we can see that 2 parameters are using in this study, which are aerobic plate count and yeast and mole count. As we know, food in fermented fish in Asia contains of mixed microbiota. It is have a mixture of the bacteria and yeast and mold. So that's why we need to perform both aerobic plate count and yeast and mold count. And from this study, we can see that during fermentation, the bacteria for aerobic plate count is going up for the first 5 days, and after that, after the prolong the fermentation time, the microbial count becomes steadily maintained for the black pepper, and then for the chili and turmeric, they have a slightly up and down. And for the yeast and mold count, basically yeast and mold count in all samples were lower than the aerobic plate count. It shows that most of the bacteria actually is more dominant compared to yeast and mold in all the samples. Okay, for the objective 3, to determine the sensory acceptance of the fermented tilapia, this is the results showing the color, odor, texture, taste, and overall acceptance. And what we can conclude in this study that the microbiology count in the aerobic plate count of fermented tilapia range from 10 to power of 5 to 10 to power 8. It shows that the fermented tilapia actually within the acceptable level, because we know that we cannot consume the fermented fish raw. That's why after definitely after cook, the amount of the bacterial count will extremely reduce and will be safe. For yeast and mold count, the 6% turmeric reduce the yeast and mold count until it become less. And for the bacterial count, all 3 samples had reduced the level of microbial count. However, the reduced microbial load is still not less than its specific safe level. But as I said, after we cook them, definitely it will reduce to the safe level. LAB count of fermented tilapia added with selected spices increased gradually throughout the 15 days of fermentation. And based on the sensory evaluation, there is no significant difference between all the samples. However, the best formulation was found for the 3% black pepper, and second best formulation was 6% black pepper. For the further study, we would like to continue this study looking at on a different packaging system, and at the same time maybe study on the different types of the spices.